The Heliconius butterflies are found in the jungles of South and Central America. Their wings are patterned with an amazing variety of bright colours and shapes. And recently they've become a great system for understanding how genes control the diversity of animal form that we see in the natural world.
Butterflies are great for studying gene patterning because they have these lovely two-dimensional wings which you can stain for different genes during development. And this will show you where and when these genes are turned on and how that relates to the final colour pattern of the adult wing.
In the 1970s and 80s, geneticists mated different species of Heliconius butterflies and observed how their offspring varied. What they found was that there was a small number of genes responsible for the control of all major wing elements. Nowadays, with modern techniques such as whole genome sequencing, we're able to find where these genes are located within the genome, and we have the power to identify the switches that turn them on or off. But first, we have to find the butterflies. And so, although not quite as intrepid as the original Victorian explorers who worked on Heliconius, we continue to return to the neotropics armed with butterfly nets. We travel to the jungles throughout South and Central America, in countries such as Peru, Suriname, and Panama, to find the specific color pattern forms that occur there. The butterflies we collect in the wild are used for DNA sequencing and for breeding experiments in the insectaries, like this one in Gamboa on the banks of Panama Canal. The wing pattern elements are defined by genes turned on in the pupa during the transformation from the caterpillar to the adult butterfly. We raise the caterpillars and pupa in the insectaries to see what the patterning genes are doing in the wings.
For example, there's a gene called optics, which is switched on in the wings of the pupa. Wherever optics is switched on in the pupal wings, the adult wings will appear red. Some butterflies have red bands on their forewings, and optics is expressed where these bands will later appear. Other butterflies have red rays, and optics is expressed in each of these rays. So the gene is the same for both of these red patterns, but what's different is the switch that controls whether optics is turned on or off. These switches are activated by different genes in different parts of the wing, and it's this that defines the pattern of optics and ultimately the pattern of red.
We're beginning to understand how some of these genetic switches work and produce the diversity of wing patterns in the Heliconius butterflies that we see today. This is part of the much bigger picture of understanding how the great diversity of animal life that we see on Earth today has evolved.